Hello everyone, my name is Andrew Kim and welcome to the 6th unit of AP Bio Review session. Now, 6th unit is about DNA replication, transcription, and translation. So now, let's move on to the very first part of this unit, which is DNA replication. Now, DNA, how does DNA look like? Well, sorry. In fact, before talking about how DNA looks like, I would like to first talk about why DNA replication is necessary. There are a lot of videos out there explaining how DNA replications work, but there isn't any video that explains why DNA replication, why transcription, why translation is necessary. So I would like to first explain why each of them is needed in our body. So if you remember the previous video or 2 videos ago, I explained something called mitosis, right? Mitosis is a process when we have one cell and we want to split that one cell into 2 identical cells, right? So now in this process, we are really splitting our cells. Why are we splitting our cells? Because we want to have more cells in our body. When we grow, when some of our cells die, we have to either replace or just make new cells to make our body bigger, right? So splitting cells is very important process of our body. And in order to split cells, we have to make identical DNA, right? Because newly growing part of our body still has to be me, right? It has to have the same DNA as all the cells in my body has currently, right? That is why replicating DNA and giving those replicated DNA to the splitted cells is absolutely important, okay? That is where DNA replication comes. Now, of course, transcription and translation are very important part, but that I will explain later and why it's needed in our body, okay? So first of all, let's talk about how DNA replication works. Now, how does DNA look like? Well, I've already explained this in the very first video of this whole series, but I'll explain a little bit deeper into today, okay? So as we all know, DNA looks something like this, right? Double-stranded. Now, if we were to zoom into here, how it actually looks like is it has this base, it has a phosphorus group, P group, and it has a nucleotide, right? Something like this. So what I'm drawing right now is one strand of the DNA. So this that I just drew is like this one strand of DNA, okay? Now then, let me draw the other strand of the DNA. Now, what's interesting is the other strand will not look like this. In fact, it's gonna look like this, okay? It's gonna look opposite. It's gonna be flipped. That is why we call the DNA structure as anti-parallel structure. We have 2 strands, and the 2 strands are heading the opposite direction, okay? And of course, for A, T would be matched, for G, C would be matched, and for C, G would be matched, right? Now, what's interesting is, as I mentioned, the 2 strands are heading opposite directions, so we want to name a certain way to call the direction of each strand, right? Are we going to call this north and this south? Well, we can't, because north and south is only for maps, right? It's for geology, not for biology. So in biology, and more specifically in DNA, how we call the direction of each strand in DNA is we use the direction of carbons. Now, all DNA bases, these pentagon, right? This pentagonal structure represent the base of DNA, have 5 carbons. The 5 carbons are carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. We have 5 carbons. And we always name the number of carbon as 1 starting from the right upper side, and we go in a clockwise direction to name all the other carbons, okay? Now, so if we were to do the same thing here, instead of naming here this one, we name this one. Why are we naming this one? Well, I mean, if you really think about it, 1 over here is equivalent to 1 over here, right? Because they're flipped. So 1 position here will be 1 position here. So if we were to name all the carbons on the right side as well, it's going to be 1, 2, 3, 4, 5 like this, right? Now, we're going to talk from down to bottom. Now, if we were to talk about down to bottom, what's the order of carbons? We see 3 first, and then we see 4, and then we end up with 5, right? That is why the left strand of DNA is called 3 to 5 direction. Why? Because if we were to read it from down to up, we see 3 first, and then we see 5, right? On the other hand, if we look at the right side of the strand, we see when we're reading from down to up, we read 5 and then we see 4, and then we read 3, right? We start from 5 and we end at 3. That is why we call it 5 to 3 direction. Now, I'm so mad because for some reason, my screen recording is stopping in every 5 minutes, and it is absolutely disgusting. It is so irritating, but I'll just continue. Okay, so going back to the explanation, as you see, on the left side, we have 3 to 5 direction, and on the right side, we have 5 to 3 direction, right? 5 to 3 direction. So you can see how the direction are different on both sides, right? So now this direction will come very important later. So for now on, just remember how the structure looks like, how they're opposite direction, and we read the direction based on the carbon numbers, okay? Now, let's talk about how replication actually works, okay? So this is a DNA structure. Now, you might think it looks kind of odd, right? Okay, first of all, let's just start from actual normal DNA structure, okay? So normally DNA looks like this, right? Now, in order to replicate this DNA into 2, how we're going to do is we're going to split this DNA like this. We're going to split this DNA, okay? And then we're going to add 2 parts like this. Then the end product will be 1 DNA here and another DNA here. 2 DNAs. DNA has replicated, right? This is what we're trying to do here, okay? So the first step of doing this is ripping off the DNA into 2 pieces or 2 strands, right? And how that happens is we use something called helicase, okay? So if you look at the DNA like this, we have something that looks like a scissor, which is called helicase, okay? This helicase will go through the DNA to sort of rip off. So it's like a zipper, right? It's going to zip off 2 connected strands into 2 sort of splitted strands, okay? Now, what's important is, unlike zippers, unlike clothes, once helicase goes through, yes, initially the 2 DNAs will be splitted, but they're trying to stick back together. These parts are going to try to stick back together, and we don't want that to be happening. We want them to just stay opened so that 2 extra strands can be made and DNA is replicated, right? In order for that to happen, we have something called SSB, singular stranded binding proteins that are stick to each side of DNA, and they hold back each strands so that the 2 strands don't get connected back, okay? So that is the whole point of SSB protein. Also, we have something called topoisomerase. Wait, what? Isomerase, okay? Now, what topoisomerase does is, since DNA is all tangled, it's very groovy, right? It's like it's all tangled. So even if helicase goes through and rips through the DNA, it's gonna tangle back. We don't want them to tangle. We want them to just neatly be just split it into 2 strands, right? So topoisomerase prevents these 2 DNA strands from uncoiling. Okay, so that is the whole point of topoisomerase. Now, again, we talked about helicase, we talked about SSB proteins, we talked about topoisomerase. Now, there are going to be a couple more factors that I will introduce, but please don't be confused. And of course, it will be confusing inevitably because I still have 3 to 4 more things that I have to introduce. But if it's confusing, I recommend you to just list up all the factors, all the players in this game and name their role, okay? It's like learning a new sport, right? There are going to be a lot of new positions that I'm going to introduce. And I really want you to know what the role of each position is, okay? So now, of course, the DNAs will have their nucleotides right like this, right? And as I've mentioned, our goal is to make 2 strands, one on the top and another in the bottom, to make 2 new strands, or 1 new strand, 1 additional strand of DNA, right? So in order to do that, we got to first start sort of making these connections, right? And we start by using another position, another player called primer. Primer is the place, or it's the position that makes this whole replication process start. This is where all the nucleotides will be replicated like this, okay? So primer is this singular nucleotide RNA. It's a singular nucleotide, singular stranded piece. It's like the smallest Lego block that will stick to the beginning of where the replication starts. Now, as I told you, DNA is anti-parallel, it's opposite direction, right? Now, let's assume the upper strand, so this strand is, let's say this strand is 3' to 5' direction. And if that is the case, then obviously the strand below will be 5 to 3 direction, right? Because the two strands should be opposite direction. Why? Because all DNAs are anti-parallel structure. Now, what's really important is primers, when they replicate DNA, they can never make their strand in 3' to 5' direction. Now, since primers make DNA in the opposite direction of the base strand, always primers should start from a 3' end. Why? Because primers always make DNA in 5' to 3' direction. Do you get what I'm talking about? So it's something like this. We only have all Lego blocks that we have are sticking upwards, okay? If this is a case, then the base where we're sticking all the Lego blocks should be in its opposite shape like this, right? So that the Lego blocks can actually stick to the base. Similar to that, all primers make DNA strands, make this additional strand in 5' to 3' direction. That is why this base strand has to be 3' to 5' direction. Do you get what I'm talking about? That is why always DNA primers stick to the 3' direction. So now, in the upper side, yes, here is the 3' to 5'. Here is the 3' direction, right? So it's 3' here and it's 5' here, right? It's like south here and it's north here, right? What about in the down strand? Where is the 3' location? Well, since as I mentioned, this down strand moves from 5' to 3', this part is going to be 5' and this part is going to be 3', right? That is why instead of primer being added here, the primer will be added here. Okay, so this is where primer will be added for the strand below. So that was a lot of explanations, but just to simplify it, the two primers will be added to each side, right? And the primers will be added at the opposite direction. Why? Because both strands are in opposite direction. And since the primer can always make DNA strand from 5' to 3' direction, the primer has to be added at 3' location of the DNA strand. And since the 3' DNA 3' location of the strand up and down are going to be in the opposite position. Why? Because the two strands are anti-parallel. Primers also have to be added at two opposite direction. One has to be added at the leftmost or 3' direction, and the other one also has to be added to the rightmost direction or also 3' direction. Okay? Sorry, that was not the most concise way to explain. I'm so sorry if that was a bit confusing. But let's move on, okay? Now, once this primer is added to the 3' direction, what's going to happen is there's going to be something called DNA polymerase. Okay, this is called DNA polymerase. It's another position, it's another player, okay? DNA polymerase will be introduced, and what DNA polymerase will do is DNA polymerase will make more nucleotides. It's actually going to replicate each nucleotide, and it's actually going to make the strand get longer. It's actually going to continue this replication process happen. Okay, so primer is like the starting point, the starting Lego block, whereas DNA polymerase is what adds new Lego blocks to the starting Lego blocks so they can replicate the DNA strand. Okay? So on the upper side, of course, the replication process will happen from left to right, 3 to 5 direction. And on the downward side, DNA replication is going to happen from right to left. But even if the direction is opposite physically, still the carbon direction, the biological direction is going to be the same because we're still replicating from 3 to 5 direction. Now, again, why are we replicating from 3 to 5 directions always? Well, because DNA polymerase can always make Lego blocks. It can always make nucleotides from 5 to 3 direction. And since it has to be the opposite, we can always, this is so complicated. I'm not explaining it in the perfect way. I'm so sorry. Now, honestly, just watch Amoeba Sisters for this unit. It's really hard to explain without visuals. Like, they have great visuals. It's going to make a little bit more sense, okay? So for now, just remember how helicase rips DNA, how topoisomerase stops the DNA from uncoiling, SSP proteins stops the DNA from sticking back to each other. Primer is the starting Lego block, and DNA polymerase is the thing that adds new Lego blocks to this structure, to the starting Lego block or primer. Now, what's important is, let's say we have something like this, okay? So primer, and DNA polymerase copied all the nucleotides like this. But once this is done, helicase will rip more of the DNA, right? So what's going to happen is new parts of the DNA will be unwinded like this. And helicase will be located here. And of course, helicase will continue ripping the DNA, right? Now, what's important is DNA polymerase will be here for the downside. And on the upper side, the DNA polymerase will be here, right? So of course, for the upper side, it's easier. It can just continue copying. But now for the downward side, what are we going to do? There's nothing left to copy this side. We got to copy this side, right? So what happens is new primer and new DNA polymerase is added to the new site where you have more protein, more nucleotides to copy. And once the two are added, of course, the replication process would continue. Now, as helicase rips through DNA, this process is going to continue. The DNA polymerase and primer will copy a certain amount of nucleotide, and it's going to go all the way to the beginning, and it's going to copy a bit more. It's going to go all the way to the beginning and copy a bit more, and that's going to happen, right? Whereas on the upper part, just this is all going to happen as a flow, as a natural flow in one process, in one stroke, sort of, right? Now, the issue is, if you look at the downward side, there are small gaps between, right? And it's almost like you have multiple fragments of copied strands, right? These fragments are called Okazaki fragment, a very Japanese name, Okazaki fragment. And these gaps in between will be filled by the last position that I will introduce, which is ligase. There will be something called ligase that fills in these gaps between Okazaki fragment. So that is ligase, okay? So again, I introduced a lot of different positions, but just try to remember each position's role and why it's needed, okay? So that is pretty much it for the replication part. Now, again, this was not the very best way to explain because it is complicated. You have to know the carbon directions. You have to know how it's opposite and where primers can be added and everything. But I hope this makes this makes a lot of sense, okay? So now let's move on to the next part of this unit, which is transcription. Now, similar to replication, before I explain how transcription works, I would like to first explain why transcription is needed. Why? Well, transcription is needed because we have to make proteins in our body, right? And when we're making proteins, how are we going to make a protein? Now, for example, let's say this muscle cell over here. Okay, you see my muscle, you see my huge bicep over here, right? I worked out, and now my body has to make my bicep even bigger. Now, if we want to make it bigger, we have to add new cells around my bicep, right? Now, when we're adding new cell, what color of cell are we going to add? Are we going to add a white cell? Are we going to add a black cell? No, I'm Asian. We have to add a yellow cell. But how do we know what color of cell we're going to add? Well, we know that by reading the genetic information in our body, right? Now, transcription, by definition, is reading genetic info from my DNA. So transcription is needed because when we're making proteins, we have to make proteins that align to our DNA, right? I don't want random white skin or black skin all over my body and have like polka dots. Like I want all of my body parts to be made following my genetic information, right? So transcription is used so that whenever we're making proteins, we make the proteins based on our genetic information. And transcription itself is a process of reading that genetic information, okay? Now, let me explain how this works. Now, first of all, we have to understand what DNA is and what RNA is. Now, again, DNA, as I've mentioned multiple times probably, is like a library. It's where genetic info is stored. However, there are so many different genetic information stored in DNA, right? We don't need every single DNA, all the nucleotides and all the information to make a protein, right? We only need some parts of it. That is why mRNA will copy the necessary part from the dictionary, okay? Whenever we're using the dictionary, think about it. When you're looking for a word, you never look at the entire dictionary, right? You only search up the word that you're looking for, and only you might just rip off, or you might take note of, screenshot, copy. You only take certain parts of the dictionary that you're looking for, right? This mRNA is like that small copy of the necessary part of your dictionary. Okay, so what mRNA will do is it will go through DNA and it will only copy the necessary part of the DNA. Okay, so let me show how that specifically works. Now, DNA is stored in nucleus, right? So of course, DNA transcription will also happen in nucleus. Now, what's going to happen is when you have DNA like this, mRNA will go through your DNA like this. And while it's going through, it's going to copy the nucleotides from your DNA. And once it copied all the necessary parts of your DNA, it's going to slide out and it's going to look like this, right? So this is essentially how transcription works. Very simple, right? You don't have to know all the ligase, DNA, blah, 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 top polymers, blah, 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 all the hard positions. It's very just intuitive, right? You have DNA and the strand just goes through the DNA and it copies all the necessary information and it comes out, okay? That's how mRNA works. Now, once mRNA has copied all the necessary information, it's a raw information right now, right? Like for example, when you're writing an AP Lang essay, you can't just literally control C and V the definitions you brought from dictionary, right? You have to change the font. You have to probably rephrase it. You have to probably, do all the citation and stuff, right? You need to modify the information a little bit. And that modification process also happens here. And how it happens is first you add something called poly A tail. A. And the reason why it's called a poly A tail is because it adds a adenine at the end of this RNA strand that you have, that mRNA has copied. Okay, so it adds this A tail at the end. And at the beginning, at the very front, you add something called GTP cap. Be aware, it's not ChatGPT, but it's GTP, okay? You add this GTP cap. Finally, you do something called RNA splicing. Now, how RNA splicing looks like is it literally splices out unnecessary parts of the RNA, and it connects only the important parts. I mean, if you think about it, for example, you search up the word beneficial in dictionary. Now, the dictionary definition is going to be so long. It's going to be like thousands of sentences. Okay, probably like 3 sentences, but still, you probably don't need the entire definition. You only need certain parts, right? That is why we're cutting out unnecessary parts and adding only the necessary parts. This is called DNA splicing. Now, what's important is DNA splicing can happen differently to even the same RNA strands, and that is called alternate splicing. And this alternate splicing is what gives genetic variety, or it gives some mutations. It gives some variety in the cells that will be made in our body. Yes, the cells, the proteins that are made in our body are still made based on the genetic information from our DNA, but there's small differences in what part of that genetic information we're going to use. Why? Because if all of our body is made out of literally same cells, same protein based on literally exactly same information, that won't make sense, right? I need a variety of different proteins in my body, right? I need muscle protein. I need what hairs are also protein, right? I need fingernails. They're also protein, right? We need different types of proteins. ATP synthase, even those are proteins. Enzymes are also proteins. So we need different types of proteins. And in order to make different types of proteins and add variety, this alternate splicing is used. Now, just terminology time. Now, these parts that are cut off are called introns. Introns are the useless ones, and exons will stay. Exons are the important parts. Now, think of it this way. Extroverted people are important. They're the people who make the world. They're the leaders of the world. Where introverts, they're useless. You don't really need them. So you can just cut out introns or introverted people and only keep the exons or extroverted people, okay? That's one easy way to remember. Now, that is how transcription works. Now, so what transcription does is it copies this genetic information from DNA or library into this one strand of RNA, which is called messenger RNA or mRNA, right? And it, of course, modifies as well, fully A tail. But now what are we going to do with this? Yes, we copied a small part from the dictionary, and we cut and pasted important and unimportant parts from the part that we copied to make this very, very valuable piece of information, right? Now, where are we going to use this piece? As I mentioned, we're going to use this to actually create proteins, right? Now, going back to first unit, where are proteins made? Proteins are made on the rough ER or the ribosomes on rough ER, right? So yes, DNA transcription happens in nucleus because DNA exists in nucleus, but once transcription is done, this mRNA will exit nucleus and it's going to go out to rough ER to actually go through ribosomes and make proteins. So how it's going to happen is mRNA is now outside of the nucleus. It's in the rough ER, and it goes through something called ribosome, which I already explained in first unit. And in ribosome, there's something called rRNA. It looks really weird. rRNA. rRNA basically provides this platform in ribosome, okay? So that mRNA can give its genetic information that it has copied to create protein. Now, specifically, then how is this genetic information converted to an actual protein? Well, as mRNA goes through the ribosome, what's going to happen is new type of RNA, tRNA, that looks also pretty weird, will come into work like this. Now, as you can see, there are 3 strands of whatever weird things that I drew, right? These 3 strands are also nucleotides. So what tRNA does is it's again like a Lego block, okay? Each always in a group of 3, a matching other group of 3 nucleotides will be brought by tRNA. Okay, there are a lot of different types of tRNA, and tRNA that has the matching 3 nucleotide set will come and stick to this part of mRNA and bring a corresponding amino acid. Okay, so It's like this. It's like this, okay? It's like Tinder again. But this time, instead of one-to-one dating, we have three-to-three dating, okay? So three girls are all like just this on mRNA waiting for their partners. And what tRNA does, tRNA is like a Tinder, okay? It brings three guys. And of course, three guys won't always, for example, the three guys tRNA brought are all like 25, 24, 23 years old, okay? But the girls are 13, 12, 13 years old. Oh my gosh, this is a crime, right? tRNA shouldn't be introducing these three guys to these group of girls, right? Then what this tRNA does is it moves its seat. Oh, is this the right place to introduce the 24-year-old guys? And you look at the girls and they're all 24 years old. Perfect. This is the place where this tRNA should fit, right? This is what it happens, okay? So tRNA with the matching codons, and codons is a new terminology, right? Codon means set of three nucleotides, okay? Matching codons will stick to the right place on mRNA and bring this matching amino acid. So what's gonna happen is for this three set, a different tRNA will be introduced. For this three set of nucleotides, another tRNA will be introduced, and each tRNA will bring their own amino acids, right? And what's going to happen is these amino acids will all be connected. And again, if you remember unit 1, amino acid is the monomer for a protein, right? So as these amino acids are being introduced and bonded to the adjacent amino acid, this whole strand of amino acid will be made, and that strand, in other word, is called protein, right? That is how proteins are ultimately made. Now, there's one important type of, like, there are a lot of different types of codons, but one important codon you have to remember is something called stop codon. Now, tRNAs will continue bringing the matching amino acid based on their codons, the set of three nucleotides, but there is a specific unique set of nucleotide that codes for stop, okay? There will be a group of girls that say stop, okay? Stop. Past me, nobody wants to be matched to guys, okay? So don't bring any more guys. There will be that stop codon. And once that stop codon is connected to tRNA, no more tRNA will be bringing codons or proteins or amino acids, okay? So that is where this production of protein or amino acids will end, okay? So that's pretty much it. Now, one important thing you have to remember is now our question is, yes, this is ideal case situation, right? But what if we have one wrongly copied nucleotide over here? What if this nucleotide is just not copied at all? What if we have one extra nucleotide copied here? I mean, our body is not an AI, right? There might be some small issues here and there. I mean, even AIs do make mistakes, but our bodies might make mistakes, right? What are we going to do then? Well, this would lead to, obviously, mutations, right? And there are three big different types of mutations you would have to remember. First, substitution. Second, addition slash deletion. And last, silent. Now, substitution mutation is when one nucleotide is copied into a different wrong nucleotide. For example, it should have been copied as G, but instead it's copied as A. That's a wrong nucleotide. This girl should have been 12-year-old girl over here, but oh my gosh, you put a 20-year-old girl over here. Then of course, the tRNA, the three set of guys that tRNA will bring will also be different, right? And of course, over here, the tRNA that brings the codon will be different, and thus the protein, the amino acid that this tRNA brings will be different, right? So substitution mutation can cause big issues because tRNA will bring wrong codons and thus wrong amino acid. Okay. Now, what about addition and deletion mutation? Basically, oh my gosh, why am I saying addition mutation is adding by mistake, copying one more protein. Deletion is opposite. By mistake, not copying one nucleotide that we're supposed to copy. Now, when this happens, for example, it's like this. We had 24-year-old girls here, and we have like say 15, 16, 15 girls here, right? Now, by mistake, I forgot to bring this 24-year-old girl here. And what's gonna happen is instead of 24-year-old girl being grouped in a group, 15-year-old, 24-year-old, and 24-year-old girls would be grouped as one group, right? Because we always need 3 people in one group. So now look at the group. It's awkward. It's weird. 2 24-year-old girls with one 15-year-old girl. That's messed up. There's gonna be some issues being introduced here, right? So this is how deletion or addition mutations can cause issues in our body. Now, what is a bigger issue? Substitution or addition/deletion? Well, addition/deletion. Now we can't 100% definitely say one is a bigger issue, but for most of the times, addition/deletion mutation will cause bigger problem in our body. Why? Because if it's a substitution mutation, only one group will be impacted, right? But when it's an addition or deletion mutation, as you can see, now a new group is formed here, so one group is being impacted, but then even the following groups will all be grouped with different people than they actually planned. So all the following groups will be impacted, so addition and deletion mutation can cause bigger issue oftentimes. Now, one hopeful case is something called silent mutation. Mutation has happened, but silent, there's no problem. For example, in this table, we have 15, 16, 15-year-old girls, right? By mistake, this one 15-year-old girl was swapped with a 16-year-old girl. I mean, that's a mutation that like there should have been a 15-year-old girl, but it's not a huge issue, right? Still, tRNA will bring the same guys because 16, 16, 15 versus 15, 16, 15 is not a big difference. Now, when we're talking from a biological perspective, let's say you needed CGC codon, but by mistake, we brought CGG codon. But still, if you're lucky, CGG and CGC codes for if, I'm not saying they do, if they code for the same tRNA codon, they will bring the same amino acid and no issue will be involved in making this protein, right? So that is the best, that's the lucky, fortunate case scenario, silent mutation, okay? So that is pretty much it with mutations. And in fact, okay, there's one last thing to talk about very shortly. It's just about gene expression, okay? Now, we just talked about how genes are expressed, right? mRNA copies our genetic information, and using the copied strand, we make proteins. That is how our genes are expressed. That is how this one cell in my bicep is yellow and not black or white, right? It's how my genes are being expressed. But now we're going to talk about other influences, other factors that can impact gene expression. Now, first is something called epigenetics. Not just literally DNA, but also the influences that you get from your surrounding. For example, the temperature, the sunlight, radioactive, substances or whatever things you interact in your daily life might impact your genetics, and that is called epigenetics. And that is why people say if you drink more milk, you will grow. I mean, genetics wise, your height is predetermined, right? Of course, but other factors, again, like sleep time, drinking milk, playing basketball, whatever things can still impact by minuscule level of how tall you're gonna be, right? So it's not always only the things literally inscribed in your DNA, but also the things that are just around you and your lifestyle that can impact your genetic expression, epigenetics, okay? Now, second is something like, this is more specific example. Now, when we're talking about transcription, remember how mRNA goes into DNA and it copies all the information, just like how we read things from library. Now, the thing is, in order for transcription to happen, something called transcription factor should be introduced, should be stuck to, should be attached to this RNA strand. Now, this might not happen if we have other enzymes or whatever other components in this region, and if those attached to the necessary sites, then you can't really, this transcription factor cannot be introduced, right? So what I'm just saying is there might be some questions regarding this, but just remember, similar to when we were learning enzymes, we learned about how there might be some inhibition, right? If, for example, this substrate wants to be stuck here, but there are other different components that inhibits through competition the binding between the substrate and enzyme, we will learn how the substrate will not do its work, right? Similar to that, the transcription factor wants to be attached to mRNA for transcription to actually happen, but that might be inhibited by other components that exist around it, okay? The last is something called histone. Now, in fact, histone is sort of part of epigenetics, okay? But basically what histone is DNA is in fact wrapped around these small meatballs like this. Okay, they're wrapped around these meatballs, and depending on how tightly wrapped it is, transcription might happen less. Might happen less. Okay, so when my body senses, oh, okay, we should stop expressing this specific gene, then that specific, the DNA that codes for that specific gene would be more tightly wrapped around histone, these meatballs, so that those part of the genes are less copied by mRNA and less expressed in our body. Those traits would normally be considered traits that are less beneficial for our growth, right? So those are, but still, histones work based on epigenetics. So epigenetics and histones, I won't say those two are two separate part of gene expression methods, but I would say histone is one way of epigenetic being happening, okay? So that is pretty much it for this unit. I know in some parts my explanations weren't the best. If you feel like you still don't understand some parts of this unit, then I strongly advise you to watch Amoeba Sisters, because for DNA replication, Amoeba Sisters have great video. I also made my video based on their video, so go have a look at those. And most importantly, don't forget to have a great day.